firstly to the organizers for the opportunity to deliver the presentation today. It's nice to be invited to this symposium. So a lot of what this revision entails is actually been facilitated by John Murphy's book, and especially the illustrations by Mike Roberts as well. Essentially, these are two genera which are known from the Mediterranean area fairly well. Most of the diversity occurs there, and recently we discovered them in Southern Africa as well. So you can see from the title slide, they're really tiny spiders, really small whitish cream gnaphosids. And we're going to just briefly run through the diversity in Southern Africa. Okay, so obviously the books that I purchased from John Murphy shortly after they came out, I bought the books, and they're very important because they redescribe and illustrate a lot of South African gnaphosids for the first time. And that is actually a key to actually resolving the identification of these small spiders. So just as a little bit of background, South Africa is one of the mega diverse countries on planet. It's got an exceptional spider diversity, about 2,300 species in 69 families. And of those, about 60% are endemic to the country. So that was actually part of a review paper we had last year in the Journal of Arachnology. And currently, the gnaphosids are represented by about 188 species in the country. So discovering these very small Leptodrassines occurred early in the 2000s when I started looking at the basis of Leptodrassex. It was a relatively undersampled type of biotope and microhabitat within grasslands. And I started finding a lot of these really small, tiny gnaphosid spiders. Obviously, the spinnerets were pretty obvious in terms of their classification that they were gnaphosids, but they had a very distinctive eye pattern in terms of a very strongly procurved posterior arrow. And as we continued sampling in Southern Africa, we found them just about everywhere, including in the canopies of these ficus forests and thickets, then also in coastal forests, in leaf litter, in Music and xeric grasslands, and even in the Okavango Delta in northern Botswana, also in grasses. And yes, even inside my house. So one of the species, I've collected about 10 specimens walking around on the ceiling at night. So they seem to have a strong affinity for me as well. But very easy to collect once you know where to look for them. So for South African taxonomists, identifying gnaphosids relies very heavily on the book or the publication by Roger Tucker in the early 1920s, 1923. He published a revision of all the Drassidae, or the gnaphosids of South Africa. At that stage, there were 4 very nicely illustrated plates with the genitalia of most of the species he treated in the paper. So those have been instrumental in us being able to identify our local gnaphosid fauna. But none of these genitalia matched anything that we found in terms of these very small white gnaphosids. So at that stage, we still had no idea exactly what they were. And then after John published his book, we noticed these illustrations of 2 species of Leptodrassex. The one Leptodrassex similar Simone, and then 2 undescribed species from Kenya that are illustrated as well. And they matched in terms of body size, relatively small body size, about 2 or 3 millimeters in length, and also specifically then the very strongly procurved posterior arrow. Also interesting, we noticed the presence of these feathery seta on the abdomen, which is pretty distinctive for that group of gnaphosids. Then looking at the definitions and the descriptive information he provided in the text, the number of characters in terms of the abdomen, the legs, and also then later we looked at the spinnerets, but all of these seem to match pretty much the spiders that we were finding in Southern Africa too. And amongst those, one of the morpho species pretty closely matched a species that John mentioned from Kenya that Mike illustrated, with this very distinct apical tegular process and a very strongly developed median apophysis on the retrolateral side of the palp. And the species from South Africa has basically the same conformation of the palp, also this very distinct apical tegular process, as well as a much shorter, but also still very distinct median apophysis. Also interestingly is that the embolus originates at the basal side and runs along a groove between the tegulum and subtegulum, eventually passing together with the apical tegular process. So a few years later, in 2009, Levy described the genus Leptopilos from the Mediterranean. It included several species of Leptodrassex and also similar Leptodrassex species. And it was defined then by the presence of an anterior hood in the female epigyne, as well as distinctly different conformation of the male pulp with not only an apical tegular process, but additional modifications of the median apophysis and an additional extension of the tegulum. And essentially, and then comparing the two species that John had described in his book 2 years previously, the one would then be considered to be a true Leptodrassex, and the other would actually belong to Leptopilos. So that already indicated that both genera occurred in Kenya as well, so an extension from the Mediterranean southwards. So looking at the Southern African species, once we started working through material from the National Collection of Arachnological, where most of the spiders in South Africa have been deposited recently, we found 4 distinct species that would be placed in Leptodrassex, but even 2 of those might represent a new genus. So Leptodrassex balrog and Catherine both have extremely long embolus, which runs also from the basal side, but it's accompanied in both cases by a membranous conductor. And then the apical tegular process is modified into a sickle-shaped kind of structure. And there's also a distinct lack of any median apophysis in the male pulp. In the case of the females, the copulatory ducts are extremely long and winding around the periphery of the epigyneal plate towards the posterior, and represented by very small spermatheca in both cases too. In terms of the 2 species that we can consider to be Leptodrassex, one is going to be named after John, Leptodrassex murphyi, and that has a very sharply curved apical tegular process, the short median apophysis, and the females have these semicircular ridges in the epigyne, which beneath are the copulatory openings. And that differs from a species from the southwestern parts of South Africa, which is only known from females, which has a much smaller copulatory opening and less sharply curved copulatory ducts on the inside. In terms of these 4 species, in the case of Leptodrassex murphyi, it's got a very wide distribution from central South Africa all the way eastwards into southern Mozambique. And the other 3 species have relatively restricted distributions. One is only along the south coast of South Africa, one only from Ndumu Game Reserve in northern KwaZulu-Natal. And the 4th species has scattered specimens all over South Africa, especially in the northern parts. Then looking at 3 species of Leptopilos that we've recognized from the material examined, there's 2 distinct species recognized by the presence of the anterior hood in both cases, very different septum in the epigyne, but both have a typical Leptopilos copulatory duct, a very short looping copulatory duct that enters a large oval or circular type of spermatheca. And as in the case of most of the Leptodrassines, the fertilization ducts in all these species are directed posteriorly, which is one of the synapomorphies of the subfamily. In the case of the males, you can see again a distinct median apophysis over here, a lot shorter than in the case of Leptodrassex, and 2 lamina, of which one is then the apical tegular process, and the other is then a tegular extension over there. And even after we submitted the manuscript to Zutaxa recently, I was working through additional material from the arid western parts of South Africa, and we discovered a further 3 species of Leptopilos from the Richtersveld National Park on the border with Namibia, another one from the Namakwa National Park, and a 3rd species from the Tankwa-Karoo National Park. And all of these are extremely arid types of environments, which differs a lot from the currently known distribution of Leptopilos in Southern Africa, which is largely restricted to savanna and grassland types of biotopes. Okay, so just to summarize the current composition of the Leptodrassinae, after our paper will be published, hopefully early next year. Leptodrassex then has 11 species, mainly distributed in the Mediterranean, but also with single species from West, Eastern, and Southern Africa, as well as the species from Mexico, which might be misplaced. Leptodrassex will now have 7 species, Leptopilos will now have 10, and Neodrassex from the neotropical region has 5 recently described species. And just on the right are some of the important publications dealing with those genera. In the case of Leptodrassex, there are in fact 2 species described from South Africa too, but neither has been collected again since its description. So they're obviously fairly rare and need to be collected again. Okay, in 2018, Guilherme Azevedo, who spoke a little bit earlier, published a paper on the systematics and evolution of the ground spiders, the gnaphosids. And in that paper, he then elevated Murphy's Leptodrassex group to subfamily, and that then included Leptodrassex, Leptodrassex, Leptopilos, and also Neodrassex, the currently represented genera. In terms of, as Guilherme said, everyone thinks gnaphosids are boring spiders, but some of them are really beautiful markings. Some of them are mimics of ants in the case of Micaria, and some quite brilliantly colored golden specimens from South Africa too. And in that description of the subfamily, he provided a series of synapomorphies, which we'll discuss in a moment. The first of those, okay, on track. Okay, the first of those synapomorphies I want to discuss is the apparent lack of tarsal or teeth on the tarsal claws. Now, when I was doing some work on Leptodrassinae murphyi, I actually discovered that this particular species does in fact have at least 4 teeth that are visible on the tarsal claws. So obviously, being listed as one of the synapomorphies of this group, this is a character which we need to study in all the genera, including Neodrassex and Leptodrassex in more detail to actually determine the validity of that particular synapomorphy, but it might have to be scrapped. And then another important aspect that we found in the case of Leptodrassinae murphyi was the presence of 2 very peculiar spigots on the anterior lateral spinnerets of the males. Now, in the illustrations by Mike in John's book, there were typically, in the case of Leptodrassinae, 4 spigots on the anterior lateral spinnerets of males, 2 piriform gland spigots and also 2 minor ampulate gland spigots. And that pattern was basically seen in the species from Kenya as well. But in the case of the second species from Kenya, which actually belongs to Leptopilos, there's only 1 distinct piriform gland spigot. In this case, if you look at a slight enlargement of that section in green, you can see that there's an additional 2 very small modified spigots in front of the anterior piriform gland spigot. So whether those actually are modified piriform gland spigots needs to be investigated further in a broader spectrum of species. Also to see then if that might have been overlooked in the illustrations by Mark Roberts, and whether that specific character is actually found more widely in the Leptodrassinae, or whether it might actually represent synapomorphy of Leptodrassex as a genus. Also, recent publications have looked a little bit at copulatory plugs in spiders. And in the case of Leptodrassex, it was quite remarkable to see how many of the specimens we actually examined had plugged epigynes. This is just one of the females of Balrog that I cleaned to point out where the copulatory openings are. And in the case of murphyi over there. And this is one of the plugs which I left in the epigyne just to illustrate it. But looking at all the females we examined in the study, of Leptodrassex Balrog and Capensis and Catherine, 100% of the females collected had plugged epigynes. In the case of murphyi, almost 80%. So it would appear that soon after the females emerge as adults, that they are mated and the males then secrete, or the females, some sort of copulatory plug to prevent further matings. Okay, so just in conclusion then, Leptodrassex and Leptopilos are actually very widespread, not only in the Mediterranean area, but actually throughout the Afrotropical region. And with a large number of records in Southern Africa for both genera, it seems that they're pretty common throughout the savanna and the grassland areas especially, but also extending to the arid zones. 2 of the Leptodrassex actually might represent new genera or new genus because of the very peculiar genitalic morphology that one sees in those 2 species. And the current distribution of the 2 genera actually reflects what's known in terms of sampling biases in Southern Africa and South Africa particularly, where the western arid areas have been very undersampled. We're still finding plenty of new species there, including some Leptopilos that we discovered recently. So in the case of Leptodrassex, it's very widespread too. It's got records not only in the Mediterranean, but also in West, East, and Southern Africa. And obviously, it's essential that we look at the task and the spinnerets of a broader range of taxa to establish, firstly, the absence of the presence of the tarsal claw teeth, as well as the occurrence of these very peculiar spigots on the male anterolateral spinnerets. So just I'd like to thank the organizers of the symposium for the invitation to present, as well as to Martin Ramirez and Guilherme Azevedo for the inputs and constructive discussion around the spinneret morphology. And the study was then funded through the National Research Foundation of South Africa. Thanks.
Thanks, Charles. And just on time as well.
Perfect.
So now we're going to move to our